the slides?
Yes, sir.
Okay. So let us continue our class today. So I think everyone had a good time in your semester break. And before the semester break, I think we were discussing and we have completed on the acetone, butanol, ethanol fermentations. And we have looked into the biochemical pathways, the growth curve, how the substrate is being utilized. The substrate means the glucose is being utilized for the production of the acetone, butanol, and ethanol. So we have seen that this AB fermentation is a bit a complex process in which a lot of metabolites are being produced. Like we have the production of acetone, butanol, ethanol. Along with that, we have the production of the acetic acid, butyric acids, and that depends upon how we tune our pathway. So we have seen that there are several junction points of the pathways, like it has a solvent phase and the acid fermentation phase, and which direction the fermentation will go. Okay, so it will depend upon how we tune our overall fermentation process. What are the media components that are there, how we maintain the different cofactors, other media components, and so on. So and along with that, there is a production of these acidic components, as well as with the solvent components. There are formation of two gases, mostly the hydrogen and the carbon dioxide gases that are being released. And we have seen that there are different sugars like the hexose or the fructose sugars, which can be used for the production of this acetone, butanol, ethanol. And we have seen what is the application of the acetone and butanol that is being produced through the fermentation routes. Okay, because most of them are used as a solvents, and they are used in the chemical process industries for production of different value added chemicals. Okay, so they acetone, butanol, they form the starting precursor, and they are chemically converted or treated into the or produced into the different value added other different chemicals. So we have looked into those things in detail, and then after that, we have seen that we are using in this case the Clostridium species. Okay, and they have the if you look into the growth pattern, their growth pattern is a bit different. Like they have different stages compared to the other microorganisms. Like they are in the vegetative stage or in the sporulation stage are there. Okay, so there are different stages of the growth of these Clostridium species, and depending upon which stage, if it is in the vegetative cell or it is in in form of spores, the type of the compound that is produced. So if it is in the vegetative cells, they are into the production of the organic acids. If it is in the form of spores, it produces the solvent. So if you are targeting to produce your solvent, that is acetone, butane, ethanol, you have to have your microorganism in form of the spores. Okay, so for that we have to optimize your media components. Okay, what are the what are the basic things that are required to make sure that we have the spore formations, right? So we have looked into those things in detail. Along with that, we have seen that what are the media components, like mostly we used the cornmeal because they are very much cornmeal, or we have the sugar, means we have the molasses. Okay, so the molasses, which contains the sugar concentration at around 6%, can be used as a carbon source. So these are the very these are so whenever we are talking of the media components, we make sure that if you see different media components can be used. But whenever until now, whenever in the fermentation processes we are discussing of the different media components, so these media components are the components which are used in the industrial scale. Okay, so if you look into the literature, if you look into the research or the scientific domain, a lot of different medias are being produced, and they are being optimized every day. Okay, by the different researchers across the world. And because there are different microorganisms, different starting initial carbon source, and so on, so there is a lot of variations. Okay, but in our classes, when we talk about the media components, and we talk about not only the media components, but along with that we also highlight the composition. So when we do that, we actually we take the data from the industrial processes for the acetone butanol fermentation, and generally in industrial systems, generally once the process is being optimized, that particular concentration and that particular the fermenter system or the overall process is like being followed for down the years. Okay, once you optimize the system. So we are in our classes, we will be highlighting those media components and those compositions and the time frame, okay, for the fermentation and so on. Okay, so that is one thing that everyone should keep in mind when we are discussing the media components. Okay. So then after that, we have looked into the growth curve, and then after, so we have seen there are this basically three phases of the growth, where we have the first phase, okay, where there is a rapid formation of the acetic acid and butyric acids, which is the acidic organic acid components, and then we have the second phase where with this acid, the pH was the almost decreased, because these acids were used for the production of the organic acids. Okay, and along with that, there is a formation of the gaseous evolution of gases, which is the hydrogen and the carbon dioxide. So it is also very much important that whenever the gases are being produced, they should be vented out from the reactor system. Otherwise, it would create some sort of a pressure within the fermentation system. So that thing also has to be taken into consideration when we do design fermenter out of it. Okay, so we looked into thinkers, since means they are different. So we have so since the AB fermentation a bit complex, where we have almost two type of organic acids and three type of the solvents, the acetone, butane, ethanol, in which the acetone and the butanol is your targeted product. Okay, and the concentration, depending upon like how much you would like to have your acetone or how much you would like to have the butanol, you can actually tune it by the addition of the different enzymes, and that we have already seen in our the biosynthesis pathway. Like for example, if you want to improve the production of your for example ethanol, you have to have a reducing environment where you add the NADH or the NADPH. Okay, and for example, if you would like to improve the production of, for example, the acetone, then we have a several enzyme, that is the ferredoxin oxidoreductase, actually, which can improve, for example, the acetone or the this acid production. So by depending upon which type of enzymes or which type of like if you maintain the reducing environment, in this case we can actually tune that your fermentation process, like if it has to go for the production of the acetone or it has to go for the production of the ethanol or the butanol, for example. So however, in the most of our so we have seen the production of the ethanol using the Saccharomyces cerevisiae. That is traditionally, if you see in the industries, that is the most followed method for the production of the ethanol. However, if you would like to, but generally when we go for the acetone and butanol production, we use the Clostridium species for that, but the formation of a very small or minute concentration of the ethanol is unavoidable. So that ethanol is being produced um, at a very small concentration, and generally it is being removed during the fermentation process, so that the ethanol doesn't have any inhibitory effects on the growth of the microorganisms. Okay, so this is the overall what we have seen, and then we have the product recovery. So we know that both acetone and butanol have their own different, they are volatile in nature and they have a different boiling points. So generally we have this type of long or tall vertical columns, which is called as beer steel, and they consist of different perforated plates, which is heated at a different temperatures, and as the media is passed through the top of these towers, what is happening, once the liquid comes in contact with the hot perforated plates, they are vaporized, and the vapors that are being generated, they are collected and condensed. Yes, and the vapor generated is different at different boiling points, right? So if for example the acetone is volatile, it is having a boiling point around 55 degrees Celsius, 55 to 60, and if the butanol is around suppose 85 to 90 degrees Celsius, what happens if you maintain these two temperatures in these two columns? What will happen, the acetone will first become vaporize at suppose 60 degrees, and whatever the vapors are being generated, that will be collected, and that will be cooled down, and the acetone, the liquid form, the acetone will be separated and collected collected. Okay, and the rest part, which is the remaining part in the the media component, they will pass to the second column where the temperature is around 85 at 90 degrees Celsius. So the second component, which is the butanol, is being vaporized, and then they are collected and separated as a separate liquid. So generally beer still are this type of long column, the vertical column, these tower type systems, reactors, where it's mostly based on the mechanism of the distillation columns. Okay, so where we carry out the separation of these volatile components. So from and then we have briefly started on the new particular biochemical, which was the glycerol production, and we have looked into a very basic introduction on what is the importance of the glycerol, and how glycerol, like we have discussed that glycerol production was actually based on the chemical synthesis route. Okay, but how now this that the production of the glycerol also means has shifted from from the chemical based processes to the biological route of route of production through the fermentation process, and that is basically the increased the precursor, especially the in the chemical process the propylene is used, and through the chemical treatments it is in presence of some catalyst, or basically we used acid catalysts which converts the propylene into the glycerol. But the increased cost of this initial starting material has now shifted towards the production of the glycerol to biological route. And also if you are suppose using the glycerol in some for biological applications, be it for the food additives and so on, then we cannot use the glycerol that is produced by the chemical synthesis route, because I am sure everyone knows the term the racemic mixture, that is the levo and the dextrorotary, right? So generally, if you use the chemical synthesis routes, then we obtain the mixture of this L and D, which is a racemic mixture. But if you use the fermentation route for the production of the glycerol, we will obtain the L-glycerol. Okay, and that is because whenever because L-glycerol is the glycerol, the L form is the glycerol or any of the amino acids, for example, or compound. The L form is the form which can be readily metabolized by the humans, right? So if you are going to use the glycerol as a food additives, then or use, for example, in the pharmaceutical industries or for medicines and so on, and then you have to use the glycerols that is being produced by the microbial fermentation routes, because that will produce the L form of the glycerol. Okay, however, the chemical synthesis will produce the racemic mixture, and the separation of this the L and D, the racemic form into the separate L and D is very costly and a tedious job. Okay, and the separation is also not 100%. So because of that, the disadvantage with this type of chemical synthesis process, a lot of so, and this is not just only to the means to glycerols. For any of the biochemicals, if you are if you are targeting for their use in the pharmaceuticals and for the food supplements in such a way that it is taken up by our the biosynthesis pathway in our human body, you need to have the L form. Okay, and that is can only be produced that are mostly produced actually through the fermentation approaches, and most of the chemical processes are generally makes these racemic mixtures. So we have looked into this thing, and we have to look into that. So we have now moved directly to like how we can produce, for example, the glycerol. And generally, if you look into the biosynthetic pathway, basically starting with a monosaccharide, there are the glycerol production can be obtained basically by three routes. Like it forms a complex between acetaldehyde and bisulfite ions, which retards the ethanol production. So we have already looked into the ethanol production pathway. So the production of the glycerol is some a modification of the ethanol production pathway by utilization of some of the additives. Like we have the bisulfite ions. There are some other additives like we have the alkali based process. Okay, and there are other methods of using the osmotolerant yeast. So one of the other approaches is to use the genetic engineering approaches. Okay, where we genetically modify the microorganisms, especially the yeasts, which can grow, which generally have a very higher growth rate, and we make it osmotolerant. So what is the osmotolerant? So generally the glycerol and the generally these glycerol compounds, they are produced at a very low quantity, and this is because the cells cannot tolerate the presence of the higher concentration of the glycerol, because they need to so we have already seen, like the glycerol is used for example as a cryo agents, where so if you would like to preserve the microbial cells for the longer duration of the time, so it helps in the prevention of the formation of the ice crystals, like when you are freezing the microbial the cell suspension, we add some concentration of the glycerol. So if you remember, we add it depends upon how what type of microorganisms we add, from 10 to 20%, 30% the glycerol additives. Okay, and this glycerol prevents the ice formation in the on the membrane of these microorganisms. Okay, so so that is during the ice formation. It helps, it helps to maintain or it helps to prevent the ice formation. But whenever the glycerol is produced during the fermentation process, and this glycerol sometimes generates high osmotic pressure, and it leads to the rupture of the cell. So we need to make develop microorganisms of yeast which can tolerate the high osmotic pressures that is being generated when the glycerol is being produced. Okay, so to make those type of the modifications on the cell membrane, generally the genetic engineering approaches have been used. So we will look into like what are the the recent most advancement or the achievements that have been taking place in the scientific domain, especially which has the shifted which has led to the shifting of chemical processes to the biological process, especially the fermentation route for the production of this the glycerol. So if you look into this genetic engineering, so genetic engineering has been quite extensively used. So what is basically what we do in this? So we will not go into details much on what is the genetic engineering, but if you're interested, please let me know. I will share some of the information on how we can genetically modify any microorganisms, which is very interesting. Like we can take a foreign DNA where the where in which the microorganisms are quite stable under high osmotic pressures at the high concentration of the substrates of the product, and we take a targeted sequence. We identify the function of that particular gene sequence, and we take a targeted sequence, and then we modify your particular targeted microorganisms with that sequence, which helps in incorporation of that particular properties into the your targeted microorganisms. And we develop osmotolerant yeast, for example production of the glycerol, and significant improvement. So after this modification, and this modification was actually successfully carried out, and it was found that this osmotolerant yeast actually can produce the ethanol as a concentration as high as 130 grams per liter. Okay, and with a yield as high as up to 63%, and productivity, if you see, it's 32 gram per liter per day. So this is the 32 gram of the glycerol being produced per liter of the media component that is being there, and per unit day. Okay, so this was the improvement. So first advancement was always obviously the osmotolerant production of this novel osmotolerant yeast, which has higher productivity. And if you see the general, after the optimization and after the media optimization, generally the glycerol yield productivities is almost 58%. I mean, if you compare with the broth, it's almost consumes it almost 58%. Okay, and this is significantly higher. So that means almost 58% of the carbon source that is being provided is actually um, converted to the glycerol, and in your in your final product that you obtain after the fermentation, the 58% of it is will be the it will be your targeted glycerol, which comes to around 30 gram per liter per day to 110 to 120 gram liter per day. Okay, productivity. So this is whenever we talk about you know gram per liter per day, that is actually basically the productivity of the glycerol, and 100 gram 110 to 100, 120 gram per liter is tells you about the concentration in the particular broth. Okay, so if you can see the values and numbers, so they are significantly higher, and it's also comparable with the chemical processes. And these values are obtained only when we have optimized, the aerobic fermentation process. So it's like for any fermentation process, if you are going to obtain, the highest yield of your targeted product, you have to optimize the several things. If it is aerobic fermentation, you have to optimize, what is the concentration of the air, or the concentration of the oxygen, and what are the concentration of the growth media components, because we have seen and discussed that the growth media components, which always contain both the micronutrient as well as the macronutrient, and some of the cofactors. So so the concentration at which they are added, okay, and what are the micronutrients that should be added, that needs to be optimized, okay. And then only we can obtain this very high concentration of your final product. Okay. Now after you have produced your, the glycerol, and it's present up to suppose 58% in the broth, we have to now separate it. So that is also a challenging task, and the particular technique called as a carrier distillation technique has been developed, which can actually distill out the glycerol as with a efficiency as high as 90%. Okay, so this particular technology has been developed. We look into that, what is, the carrier distillation, and how it is performed when we discuss the downstream processes. So but however, you can see that at each and every step a significant, the scientific advancements have already been made. Okay, and that has come from the research that has been carried out, especially in development of the novel strains, in development of a proper downstream processes based on the carrier distillation approach, and proper optimization tools to define statistical approaches, so that we obtain the very high concentration of the productivity of your targeted, biochemical, which is the glycerol in this case. So this has been, the scientific advancements that has been done till date, and we will see what is the phosphotolerant yeast, what is, the bisulfide base, as well as the alkali based processes, and what are their, the advantages and the different assays of all these steps. Okay. So before that, let us look into briefly of, the glycerol is being produced. Okay, but after production, what is the application of it? So we have to, because a particular biochemical or the demand of that, it will only come when they have a very high performance, the application in different domains. So if you see the glycerol, a chemical name is basically 1,2,3 propane triol, because it is having, three groups. So it is a form of alcohol. It's a simple alcohol, and with many uses, especially in the cosmetic paints as additive. Okay, the automotive, food, pharmaceutical, pulp, paper, later textile industries, or as feedstock for production of various chemicals. That's okay. So the glycerol has a lot of, application and uses in the different sectors. Now if you are going for the automotive, or sorry, if you're going for the food or the pharmaceutical industries, generally the desired form of the glycerol will be the L form. So as we discussed in the previous two slides, that in that case it is very specific that we should obtain the L form of the glycerol. But if it is being used for other applications where, the biological interference is not there, like in the cosmetics, paints, or for example the pulp and paper, leather and textile industries, in those industries the D form is also can be used, or also the L and D both are applicable. So we obtain the generally go for the racemic, the racemic mixtures. Okay, and during those things, if it is for that particular applications, generally the glycerol is being produced through the chemical synthesis route. Okay, and if you are for going for the biological, like the food or pharmaceutical, we go for the biological route or the fermentation based route for the production of the this glycerol. So some of the examples, if you see what products has been made within the glycerol, especially in the chemical industries, is like with the glycerol, it can be fermented to produce 1,3-propanediol. Okay, and this propanediol actually can form, feedstock for the production of trimethylene terephthalate. Okay, and this particular new polyesters, which are basically used in the fiber and textile applications, they possess excellent properties like very high strength, okay, good resilience, inherent, strain resistance, and low static generation. Static generation means the static charge generation. Okay, generally plastics, if you see, they if you take a new plastics or polythene bags, you can feel there is a formation of those charge. So however, if we for example produce this particular, the polytrimethylene terephthalate, they are very strong with good resilient properties, and they generally have a low static the charge generation. Because of that, it can be used, for example, making the body components or parts of any electronic products. Okay, so if so that is very much important. So whenever we select any material for making different products, be it the electronics or be it in the food additives or any other industries, so we have to look into that characteristics of that particular product, and then we have to decide. So like if we are going for the food and pharmaceutical, we have to go for the L form of the glycerol, and for that we need to have we need to produce for that we need to produce the glycerol through the fermentation route. Okay, next, if we look into the bacterium for the production of the glycerol. So generally, the glycerol production takes place by the transformation of the glyceraldehyde 3 phosphate to the glycerol phosphate, and that is converted into glycerol. But the glycerol can also be further converted into another product which is called as a dihydroxyacetone, and this dihydroxyacetone have several applications. And they have this dihydroxyacetone is used as a precursor for production of different type of the polymeric additives and so on. So, and this conversion of glycerol to the dihydroxyacetone can actually be catalyzed by some of the chemicals with catalyst, or it can also be carried out by the bacterium called as Acidobacter suboxydans. Okay, so glycerol can actually act. So this is the very important, that it acts as a feedstock for the production of various chemicals. Okay, so if you go for the dihydroxyacetone or for any other chemicals, it can be a very good initial starting material, and then it can be converted into various chemicals by both chemical approaches or by the biological approaches. Okay, and if you look into the fermentation type, okay, so if you look for the conversion of this glycerol to the dihydroxyacetone, generally it is carried out through the submerged fermentation, and the bacteria almost produces with a yield of 75 to 90%, with a starting concentration of the glucose with 5 to 10%. Okay, so being the 5 to 10% of the glycerol, from that almost 90%, 75 to 90% of it is being converted into the dihydroxyacetone, and this dihydroxyacetone further is being transformed into the dihydroxyacetone kinase or dihydroxyacetone phosphate, and they act as a substrate to make the optically active sugar derivatives. So some of the sugar derivatives, such as some of the sweetening agents that are being used. Okay, so they are basically nothing but this type of dihydroxy acetone phosphates, which can be produced from the glycerol. Okay, and using the microorganism Acidobacter suboxydans, and generally it is obtained at a very high yield. So yield of 75 to 90% for any biological process, okay, is considered to be very high. Okay, but it is obvious that in the chemical synthesis process we may get the yields of as high as 99%, but for any biological processes, yield of 75 to 90% is very much is considered to be high enough. Because what is happening, whenever you are providing a glucose as a carbon source, some of that carbon source is is utilized by the microorganisms for its metabolic activity. So what is the metabolic activity? Metabolic activity represents the daily means to grow the cell number, to maintain the cell for its cell growth and so on. So some of the energy is used for that, and rest of the energy that is there in the glucose carbon source is used for the production of your particular, the metabolites, or for in this case the glycerol. So if you are having, so that means if you are getting an yield of 75% of your final product, that means around 15% of the carbon that is there, that has been used up as a for to maintaining the metabolic activity of the cells. Okay, and that is very, this particular metabolic activity is very much important. So if the cells are metabolically active, then only the products will be produced. Okay, so for any fermentation process, given the carbon source, some fraction will be used up for maintaining the metabolic activity, and then some fraction will be converted into the product. Okay, either it can be converted directly from the carbon source that is there to the product, or it can be used up by the microorganisms, and then it will be released in form of some of the products. Okay, so this is the general, the pathways, general, that information on the glycerol, and how the glycerol derivatives can be converted. Glycerol can be converted into different derivatives, and they are used for the different applications, like in this case sugar derivative, for the production of this particular polymer, which is having high strength and resilience, and so on. And also, if you are using a glycerol from the biological source, for the production of this type of any biochemicals, it will be considered to be as a environmentally benign. That means it should be considered as a means it will lead to generation of the lower, the greenhouse gas emissions and so on, because whatever the carbon atoms that are present in this glycerol backbone, those are obtained through the biosynthesis route. Okay, and not from the chemical synthesis routes, which generates a lot of, the greenhouse gases and so on. So if we follow these things along with, the improved properties for the products, we also make overall our process as a sustainable process. Okay, now let us look into the different process that we talked about. So there are basically four processes, and if we consider with the yeast, okay, so either yeast are used generally for the production, or some of the algal biomass are there, which can be used for the production of, this glycerol. And if we compare the three processes that are there, that are the sulfide process, the second is called the alkali process, and third, by the genetic engineering approaches, when we modify the cell membranes to make it osmotolerant, then we call it as a osmotolerant yeast process. Okay, so if you see the microorganisms, generally we have, in the case of the yeast process, we have for the sulfide and the alkali, we have the Saccharomyces cerevisiae, and the Saccharomyces cerevisiae is also, we have seen that it is used for the production of the yeast, as well as the biomass production of the ethanol, as well as the biomass that is being generated, that is also used as a as the additives to the baking powder, and it forms a baker's yeast. Okay, so same microorganisms can be used for, the production of the glycerol. And if we look into the biochemical pathway, so we'll look into this into more detail. Like, so it starts with initial glucose as the carbon source, and that glucose will be converted to, the glucose 6 phosphate, and this glucose 6 phosphate will go into the glycolysis pathway to the fructose 6 phosphate and to the glycerol phosphate pyruvate, and produce ethanol and carbon dioxide. So everyone, I am sure by now everyone, we have already gone through this pathway where we convert the glucose to the ethanol, and it leads to the generation of the carbon dioxide. Okay, and so on. But in this case of the glucose production, glycerol production, different pathway has to be taken, and that takes at this particular third step of this glycolysis pathway. So in this case, what is happening? Whatever the glyceraldehyde that is being 3-phosphate that is being produced, so or the fructose 6-phosphate that is there. So this fructose 6-phosphate is converted into dihydroxyacetone phosphate, or if the glyceraldehyde 3-phosphate is being produced for the glycolysis pathway, it is being converted into this DHP. Generally, it's called as a DHP. It is dihydroxy acetone phosphate. So both of from the both the sides, we make sure that we obtain the high concentration of dihydroxyacetone phosphate if we are targeting to produce example the for example the glycerol. So this dihydroxyacetone phosphate is converted to the glycerol 3 phosphate by the using the enzyme GPDP. So GPD is the glycerol of phosphodehydrogenase, phosphate dehydrogenase, and this one so here we have the phosphodierogenase which introduces the phosphate group into the backbone, and then we have the glycerol pyrophosphate where it removes this particular phosphate group and we obtain the final glycerol as a product. So this is a very simple pathway, you can see, compared to the ABE fermentation. This is a very simple pathway where at the step 3 of this glycolysis cycle, okay, what is happening, this fructose 6,1,6 biphosphate is converted into a dihydroxy acetone phosphate, or the glyceraldehyde 3 phosphate is converted into DHP, and then that the DHP is being converted to the ethanol. But one thing to look into is that both this process, the production of the glycerol and the ethanol actually goes hand in hand, and generally there is a production of some quantity of the ethanol is also produced during the fermentation of the glycerol. Okay, now we have looked into this biochemical pathway. Okay, now we will look into that like there are three processes like the sulfide process, the alkali process, and the osmotolerant. So as the name suggests, the alkali process is like we need to require to add some sort of alkalis, and in the case of sulfide we require to add in some quantity of the sulfide. Okay, and this addition is going to change the is going to tune or affect the biochemical pathway in such a way that it will improve the production of the glycerol. So we will keep our class until now today, because I have a the meeting to attend. So let us discuss this particular part on how the presence of this bisulfite or the sulfide ions or the alkali process actually alkali actually affects the overall pathway. So the presence, so there is this aldehyde molecule that somehow is interacting with the sulfide ions. So that is how that is responsible to form the acetaldehyde bisulfite compound, which helps which is why we need to add, so which is why we need to develop a different process called as a sulfide process, and similarly it goes with the alkali process. Okay, so we will discuss this in detail in our the next class. Okay, so until now, anyone has any questions or doubt? Hello, hello, am I audible to everyone?
Yes, sir.
Hello.
So,
Okay, so okay, so let us keep our class until now today, and we'll discuss more on the next class. Okay, so thank you everyone for joining.
thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.